Assalamu alaikum. My name is Dr. Abdullah Saghir Ahmad and I am working as an assistant professor in the Department of Parasitology, Cholistan University of Veterinary and Animal Sciences, Bahawalpur. I am working as an assistant professor since 2018 and today I am going to discuss the tick-borne diseases of canine. In this study, we find out the most prevalent diseases in canines like babesiosis, ehrlichiosis, and hepatozoon. The most important thing about these tick-borne diseases is these are emerging zoonotic diseases, and the animals remain carrier throughout of his life, and most prevalent. And till now, not a single molecular study has been reported from Pakistan. That's why we did this research. Here is the GS map that represent the three different localities for in the study. These areas were selected on the basis of geographical distribution and their agroecological zones like Muzaffargarh, Kasur, and district uh, Rawalpindi. Here is the primer that we used for the diagnosis of Babesia gibsoni and Babesia vogeli for the molecular confirmation, and then we perform the Sanger sequencing for their sequence analysis, and then we did the phylogenetic analysis of Babesia gibsoni. In this picture, you can see the hours samples that represent the Babesia gibsoni from a single clades that are most related to other relevant countries like Spain, China, USA, India, and Japan. Then we did the phylogenetic analysis of Babesia vogeli, and we found that our samples found two different clades with different localities like India, Brazil, North Tunisia, Spain, and USA. After that, we did the phylogenetic of Ehrlichia canis, and we found that all the samples of Ehrlichia canis form a single uh, clade with Taiwan, Philippines, Cyprus, Brazil, and Kerala, India, Japan, China, Brazil again, and Italy. And one of the sample is most our that we found in our study is totally different and form a different clade. And the last phylogenetic analysis that we perform is the hepatozone canis, and hepatozone canis form the four clades. Three are from our studies, and rest is the other reported studies. We use the Toxoplasma gondii as an outgroup for the representation of the uh, phylogenetic analysis. Assalamu alaikum. My name is Dr. Abdullah Saghir Ahmad and I am working as an assistant professor in the Department of Parasitology, Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Today, my topic of discussion is the ticks and tick-borne diseases of canine, in which we discuss the most important tick-borne diseases like babesiosis, ehrlichiosis, and the hepatozoon canis. Babesiosis are the most important zoonotic diseases, and the animals remain carrier throughout of his life, most prevalent, and not a single study, molecular study, has been reported from Pakistan till now. Here is the map that represents the three different geographical locations that from where we collected the samples for the diagnosis of these three important diseases, namely the district Muzaffargarh, district Bahawalpur, district Rawalpindi, and the district Kasur. The primer that we use for the molecular diagnosis of the Babesia gibsoni and Babesia vogeli are mentioned here. And after the molecular confirmation, we go for the Sanger sequencing for the molecular analysis and bioinformatic analysis of these prevalent species. After the bioinformatic analysis, we did the phylogenetic analysis of Babesia gibsoni, Babesia vogeli, Ehrlichia canis, and the hepatozone canis. Here, the first one, the phylogenetic analysis of Babesia gibsoni. The species that we found in Pakistan are making two clades closely related to China, Spain, USA, India, and the second clade is totally different, Babesia gibsoni 11, that shows in the picture. And here we use the Plasmodium falciparum as an outgroup. The second phylogenetic analysis of Babesia vogeli. Here we can see the five species that we found in our studies are making the three different clades, and the Neospora caninum were used as an outgroup. Phylogenetic analysis of Ehrlichia canis. This picture depicts the our samples from two clades and only the E can 2 and 3 making a separate group while the remaining are making the same groups with the Taiwan, Singapore, Brazil, Cyprus, India and China, Greece, Italy, while the Rickettsia were used and an outgroup. The last one is the phylogenetic analysis of Hepatozone canis. In this study, we found out the our samples making the three different clades with the reported sequences like Thailand, West Indies, Pakistan, Taiwan, Portugal, Italy, Brazil, and Spain, while Toxoplasma gondii were used as an outgroup. Thank you.